In this video, we are going to finish the protein structure, and then we are going to talk about the last group of organic molecules, which are the nucleic acids. In the last video, we were talking about the peptide bonds, and we saw how through dehydration synthesis or condensation reactions, we can form a peptide bond, and then through the reverse reaction, which is the hydrolysis reaction, we can break down a peptide bond to get the forming two amino acids. At the beginning of this video, we'll be discussing the hierarchy of organic protein molecules. We said before that in order for a protein to be functional, it has to be in a three-dimensional shape or in a three-dimensional conformation or in a three-dimensional configuration. So all these are equivalent terms that you can use in order to describe the shape of a protein molecule. So when we say conformation or when we say configuration, then we mean the shape of the protein molecule. Because the protein molecules are made of amino acids, and because these amino acids can be like arranged in a different number of amino acids in the same chain and in a different arrangement. So this linear polypeptide sequence, we call this a linear polypeptide sequence. The term PP stands for polypeptide, and polypeptide sequence means sequence of amino acids that are bonded together through these peptide bonds in order to form a polypeptide chain. So this is an acronym that stands for polypeptide. So if you have a polypeptide chain like this one, we call this the primary structure of proteins. And this is the basic building structure of proteins. You have a group of amino acids. For example, in this case, we have 1, 2, 3, 4, 5 amino acids, or even more and more. You can have very long polypeptide chains, and these amino acids are bonded via peptide bonds. The number of amino acids can differ, the type of amino acids can differ, and the arrangement of amino acids in a polypeptide chain can differ. And that's why we have a multiplicity of possibilities of polypeptide chains. This is the primary structure of proteins. Of course, this is just a diagram, the one you can see here at the left top of the screen. But here we can see the amino acids. This is the diagram. And then here above, you can see the chemical structure of the amino acids and how they are linked together via the peptide bonds. So between amino acid number 1 and amino acid number 2, we can see this bond, the C double bond O and H. This is the first peptide bond. What about this carbon atom? This is the central carbon atom of the first amino acid. And this is the central carbon atom of the second amino acid. And then we have a second peptide bond between the second amino acid and the third amino acid, whereas this one is the central carbon atom of the third amino acid. And then this is the central carbon atom of the fourth amino acid. And in between them, we have another peptide bond. So we can see that in order to form a polypeptide chain that's made up of 4 amino acids, we have in between the 4 amino acids, we have 3 peptide bonds. Of course, for every peptide bond to be formed between 2 amino acids, we have to withdraw, or we have already withdrawn 1 molecule of water. And therefore, in order to form a polypeptide chain that is formed of 4 amino acids, we can deduce automatically that we have in between them 3 polypeptide chains, and that those 3 polypeptide chains have been made by removal of 3 molecules of water from the medium of the reaction. Of course, again, if you want to do the reverse, if you want to break down this polypeptide chain into its component amino acids, the 4 amino acids, you can again add the 3 molecules of water, and in such a way, the 3 peptide bonds will be broken, and the 3 molecules of water will be added again to the amino acids, and they will be separated. What's interesting about the polypeptide chains is that the polypeptide chains of amino acids can be very long and can be formed of repeated amino acids, for example. This is the primary structure. So here, for example, we can see the primary structure. So this is a polypeptide chain in the primary structure, and you will always have on one end a free carboxylic group, and on the other hand, you have a free amino group. Because as you see here in this arrangement, the amino acids are arranged the same way and the same direction, so that every amino acid will be placed in the polypeptide chain, so that it's, for example, here, its amino group will be to the left and its carboxylic group will be, or what remains of the carboxylic group will be to the right. So at the end of the day, at the end of the chain, you will have here a free carboxylic group if we add again the water, and here we will have another free amino group. So at the end, but this can only be seen when we look at the end of the peptide chain. So here we have the free carboxylic group, and there we have the free amino group. Of course, here you can see a complete amino group, but here you cannot see a free amino group because the molecule still continues, which means that if you look at that carbon atom, there is one bond here, one bond, a single bond, and 2 bonds here. So there is still a bond, which means that this is not the end of the chain, or otherwise we would put an OH here, and this would be a complete carboxylic group. The second level of complexity of proteins in the hierarchy, and when we say the hierarchy here, we mean the level of complexity of the structure of the protein molecule. After the primary level, we have the secondary level. In the secondary level, we just take the same polypeptide chain, a single polypeptide chain, and we fold it. But we can either fold it as a helix in a helical manner, like this, in which case it is called a helix. And alpha helix determines the way it is rotated. Is it clockwise or anti-clockwise? But this is not important. You're not supposed to know what alpha is and what beta is. You just have to know that in the secondary level, we have this single linear polypeptide chain, but we fold it as a helix. In the beta-pleated sheet, the polypeptide chain will also be folded, but in rather acute angles like this. So this is a molecule, and this is another molecule. So they are 2 different molecules. They are just giving you examples of 2 different examples of the secondary structure of proteins. One of them showing the first possibility, which is the helical structure, and the second one showing the beta-pleated sheets. But in both, in each of these 2 molecules, we only have one polypeptide chain. And it's worth noting that until the tertiary structure, we just have one polypeptide chain that is further folded and folded. So the main difference between the primary structure, the secondary structure, and the tertiary structure is in how much folding there is. The least folding occurs in the primary structure, of course, because it is already non-folded. It is linear. In the secondary structure, you have some degree of folding in a helical way or in a pleated way. But in the tertiary structure, this is still more and more folded. So we have more complexity, and that further folding and that more complex structure makes the tertiary structure three-dimensional. And once it is three-dimensional, it has a particular 3D shape in space. And therefore, you can have, for example, concavities or depressions on the surface of the molecule, like we saw, for example, in our study of enzymes, when we saw that this depression was the active site of the enzyme. And that's why this led scientists to deduce that in order for a protein molecule to be functional and to be able to exert a particular function in the human body, it has to be at the 3D level. So up to the tertiary structure, we just have one polypeptide chain with the least complexity at the primary structure and the greatest complexity at the tertiary structure. Of course, you might be wondering how the alpha helix stays fixed in place. Why does it stay helical like this and it does not fold? Or how does the beta pleated sheet stay pleated like this and does not unfold? This is possible only because in order for these folds to be formed, there are specific bonds that are formed between specific atoms in the chain so that it cannot unfold. The same applies to the tertiary structure. So when you move from the primary structure to the secondary structure, this means that folding has occurred through formation of new bonds that fix the atoms in place to keep the molecule folded. And we will see this in further details on the next slide. Similarly, when you move from the secondary structure to the tertiary structure, this means that you have made new bonds that caused the secondary structure to be more and more folded. The same applies to the difference between alpha helix and beta pleated sheet. Why would a molecule fold into an alpha helix and not into a beta pleated sheet? This all depends on the type of new bonds that have been made for further folding. And this is what we are going to see on the next slide again. On the contrary, for the quaternary structure, this is the first level of hierarchy of protein molecules where the molecule can contain more than one polypeptide chain. So this is an MCQ question. So if you're ever asked what is the level of protein hierarchy where the molecule of protein contains more than one polypeptide chain, you would say the quaternary structure, whether this quaternary structure includes 2 polypeptide chains or 3 polypeptide chains or more. All this makes the molecule in the quaternary structure. Now, here we are going to discuss first the primary structure again. And as we said, it is a linear sequence of amino acids. And it is a unique sequence of amino acids, as we said before. Because, why is it unique? Because it depends upon the type of amino acids that share in the formation of the chain, the number of amino acids, as well as the order or the arrangement of the amino acids in the polypeptide chain. If you make any slight change in the type or number or arrangement of the amino acids in this chain, the slightest change can have major consequences not on the structure, because you have changed only one amino acid, for example, but on the function of proteins. of this particular protein where you made a change. So here we can see, for example, that you all know, of course, that if we have a double-stranded piece of DNA, every 3 consecutive nitrogenous bases on the DNA strand will determine 1 amino acid. So we can, on top of DNA, form RNA through complementary base pairing, as you might have studied in DNA in your first year of high school or in the middle school. So, for example, complementary to C, which is cytosine, we have guanine. Complementary to cytosine, again, we have guanine. Complementary to thymine, we have we are supposed to have uracil on RNA and not adenine. So this is not the strand that was used for transcription of RNA. So I would repeat again. So in order to use a sequence of DNA to form a sequence of RNA, a process called transcription is used, and that process of transcription, of course, requires transcription enzymes and requires transcription proteins. So it's obvious that So I would repeat again because there was some kind of ambiguity. I apologize for the mistake I made. So this is the double-stranded piece of DNA, and it consists of 3 nitrogenous bases. So every 3 nitrogenous bases on DNA represent, which we call the genetic code, will be transcribed via a process of transcription that requires transcription enzymes and transcription proteins into a sequence of nitrogenous bases on RNA, which we call a codon. So this triplet on DNA, the sequence of 3 nitrogenous bases on DNA, we call a triplet code, a triplet code. So in this example, we have used this triplet code on DNA to form the complementary codon on RNA. So complementary to cytosine, we have guanine. Complementary to thymine, yes, it's okay, we have adenine. There is no mistake, there is no problem. And complementary to cytosine, we have guanine. This codon represents an amino acid that is called glutamic acid. GLU stands for glutamic acid. So if glutamic acid is included in the polypeptide chain of hemoglobin that is present in the red blood corpuscles, then this is a normal molecule of hemoglobin. And the protein we are talking about here is hemoglobin that is present inside your red blood corpuscles. But it happens that there might be a mutation, and that mutation we call a point mutation because we have only 1 nitrogenous base that has changed. So instead of having adenine here, we have thymine, and it is written in red because it's not the original base that should be present. And therefore, here in front of it on the opposite strand of DNA, we don't have thymine, but we have adenine. So instead of having thymine here, we have adenine here. And that results in a difference in transcription because when you use this sequence, which is CAC, and transcribe it into a sequence on RNA, which is a codon, you will have a mutated codon, which has GUG instead of GAG. So it's only one difference in the codon, but this time GUG will not be translated into the amino acid valine, will not be translated into the amino acid glutamic acid, but it will be translated into the amino acid valine. So what does this mean? This means that if we have a single nitrogenous base changed on the sequence of DNA, which we call a point mutation, this will result in a change of a single nitrogenous base on RNA, and this will give you a totally different amino acid. So instead of having glutamic acid, you will have valine. Again, the use of DNA sequences to form RNA, this is a process that we call transcription. And the use of this sequence of RNA to form a sequence of amino acids, this is what we call translation. And every amino acid in your polypeptide chain will be represented by a sequence of 3 nitrogenous bases on RNA, which we call the codon. And if you haven't already studied DNA and RNA and nucleic acids, this is no problem, because we will be studying this in full detail in this section and in the section on molecular biology. Of course, if you replace glutamic acid by valine, then the protein is now a mutant protein. So instead of having normal hemoglobin inside your red blood corpuscles, you will have a mutant hemoglobin, or you will have abnormal hemoglobin inside your red blood corpuscles, and this results in a disease that is called sickle cell anemia. Why is it called sickle cell, and why is it called anemia? This is because these red blood corpuscles, suppose this is the red blood corpuscle, and suppose that it contains the mutant hemoglobin or the mutant protein. So upon exposure to low oxygen levels, for example, upon heavy muscular exertion, when the muscles withdraw oxygen from the blood, this protein inside that is originally globular will change its shape upon being exposed to low oxygen tension and will be unfolded. And this unfolding results in a change in the shape of the red blood corpuscle. Instead of the red blood corpuscle being spherical, the red blood corpuscle having this mutant protein inside, this protein has been now extended, unfolded, extended because it has been unfolded, and the red blood corpuscles change their shape, and instead of being spherical, they become sickle-shaped. You know, the sickle-shaped, this looks like this, very similar to the tool with which we cut the grass, and that's why it is called sickle cell anemia. Okay, we have understood why it is called sickle cell, because the red blood corpuscles become sickled upon exposure to low oxygen. But why anemia? Because these elongated, deformed, sickling, of course, is a deformity. So they are now deformed red blood cells or corpuscles. And these red blood corpuscles that are deformed will clog the blood vessels and will result in occlusion of the blood vessels. Therefore, blood does not reach the cells, and this might result in many symptoms and also in death of the individual. The first scientist who sequenced amino acids or sequenced proteins in general was the scientist Fred Sanger. and he received the Nobel Prize for having sequenced the famous polypeptide hormone, which is insulin. So in the primary structure, we just have peptide bonds between the amino acids, as we said before. But in the secondary structure, how does this folding of alpha helix and beta pleated sheets occurs? In the secondary structure, this is local folding. Folding is understood. But before saying this, I haven't yet mentioned what anemia is. Coming back to the sickle cell anemia, when the red blood corpuscles are deformed like this, they clog the blood vessels. This is one problem. But the second problem is that these red blood corpuscles rupture. They get ruptured and they release their hemoglobin outside. And therefore, you have fewer red blood corpuscles and the red blood cell count decreases. So the red blood cell count decreases, and this is what we call anemia. And that's why it is called sickle cell anemia. Now we come to the secondary structure. In the secondary structure, we have local folding within a primary polypeptide chain, as we said. So we don't have two different polypeptide chains, but it is a polypeptide chain where we made local folding. How do we do this? Or how do we make these bonds? By having hydrogen bonds between the oxygen of the carbonyl group of one amino acid and the hydrogen, the oxygen of the carbonyl group of one amino acid and the hydrogen of the amino group of another amino acid. So if we come back to this, we would find that where is the central carbon atom here? The central carbon atom is here. And this is the peptide bond. So here we have a carbonyl group, and here we have an amino group. And then if you look here, this is the hydrogen bond we are talking about. And if you count the number of amino acids, so if this is, for example, the NH of the first amino acid, so we would have here a central carbon atom that hasn't been put on the diagram. So this is number 1, this is number 2. And then this carbon atom is not the central carbon atom. It is the central carbon atom of the carbonyl group. This is the third central carbon atom of the third amino acid. And then this is the fourth central atom. Let's count again because the diagram is very confusing. So we are supposed to have this bond between the oxygen of the carbonyl group, as we said, of one amino acid and the hydrogen of the amino group. And in order to have the alpha helix we were talking about, it has to be 4 down the chain. It has to be the 4th down the chain. That's why we are going to count again in order to see that this bond is between this hydrogen that is partially positive and this oxygen that is partially negative, and we have the hydrogen bond in between. So if this is the first carbon atom, as that is supposed to be here, then this one should be the 4th down the chain, and we will see how again. Let's count again. So if this is the first central carbon atom, so this would be the 2nd carbon atom, this would be the 3rd carbon atom, and then the 4th central carbon atom, and this is the 5th central carbon atom. It is the 5th carbon atom from the beginning, but it is the 4th down the chain. What does this mean? That when we decide where the bond will be, we will not count the first carbon atom. So this is the first down the chain. The first down BNH, and then the second down, and then the third down, and then the fourth down. Of course, we will never ask you about these details, but in order for you to understand how this folding has been done and that it is done in a special order, what's most important to understand, first of all, is that this folding into a alpha helix occurs via hydrogen bonds. And just like any hydrogen bonds, we would have a partially positive hydrogen on one side and a partially negative oxygen on the other side, and that the partially positive hydrogen comes from an NH group of one peptide chain, and that this oxygen comes from a carbonyl group of the amino acid that comes fourth down the chain. Of course, if you make these bonds, then you're going to force the peptide chain to be folded. In case of the beta pleated sheet, it is still a one polypeptide chain. Don't think because you have two segments here, this first segment and that second segment, that they are two different polypeptide chains. No, it is still one polypeptide chain that is folded like this. So this is a segment of the molecule of the chain, and this is another segment. And again, we have those hydrogen bonds. So if we read the slide, then we would say that this folding results in one of two conformational structures, the alpha helices, where you have the carbonyl of one amino acid, hydrogen bonded. And when we say hydrogen bonded, we mean through a hydrogen bond to the amino H. And when we say amino H, we mean H in an amino group, but of course an amino group that lost an H, and the amino acid is fourth down the chain. In the beta pleated sheets, we have segments, as we said, of polypeptide chains. They are not multiple, of course, they are segments of one polypeptide chain that line up next to each other, forming a sheet-like structure through these hydrogen bonds, but always between C double bond O, where the oxygen is partially negative, and H of the amino group, where the hydrogen is partially positive. And now we are going to discuss what we call fibrous proteins and globular proteins. And these are mainly related to the different shapes of proteins in the secondary structure, or sometimes even in the tertiary structure. So I think we'd better see the tertiary structure first and then study these conformational changes. But let's follow the order of the PowerPoint to avoid ambiguity. So in general, of the overall structure of proteins, whether they are tertiary or quaternary, is that they might be fibrous or they might be globular. The fibrous proteins are proteins where the polypeptide chains are organized in a parallel way along a single axis, producing long sheets. So whenever you have polypeptide chains like this that are arranged parallel to each other around or along a single axis like this, then it would be a fibrous protein. We have always told you that fibrous proteins are long, elongated molecules of protein. We gave examples like elastin, for example, in the skin, like collagen in the skin, and it's obvious that these fibrous proteins, or the polypeptide chains in the fibrous proteins, might be primary chains, or they might be secondary helical shapes that are still parallel in shape, or for example, beta pleated sheets like this. So the main criterion to say that this is a fibrous protein is that you have several polypeptide chains that are arranged parallel to each other along a long axis or along the same axis. What's the importance of having a fibrous protein? Fibrous proteins have general characteristics that are related to this parallel arrangement, because as we said in the fatty acids, when you have this rather straight arrangement, straight and parallel arrangement of the molecules, there are very small spaces in between the molecules, and therefore this fibrous arrangement allows the protein to be mechanically strong and to be less soluble in water and to be a structural protein, meaning that it's not, of course, giving structure is a function. But when we say structural protein, this means that this protein shares in the building up of the cell. So all fibrous protein, all structural proteins are essentially functional because giving structure is a function, but the reverse is not true. For example, an enzyme is a functional protein, but it's not necessarily a or it's not at all a structural protein. So all structural proteins are essentially functional because giving structure is a function, but all functional proteins are not structural because, for example, enzymes are functional, but they are not structural. And we are giving examples of structural proteins. So examples include 4 major examples. We have alpha keratin, we have collagen, as I said, we have wool, and we have silk. They're all fibrous proteins, but there are differences between them. We see alpha keratin consists mainly of alpha helices, as the one we have seen on the previous slide. So you would have several alpha helices that are arranged parallel to each other, and this is alpha keratin that we find in hairs, in nails, in claws of animals, in beaks, and in scales. So this is a famous example we have studied in our study of the skin and the integumentary system. And then we have collagen, and as we know, collagen is present in the tendons. Every muscle is attached via a connective tissue tendon to the bone where it is attached. So collagen is not only present in the skin, as we all know, but it's also present in bones, it is present in the teeth, and it is also present in the tendon, and it is made of helices, but these helices are non-alpha helices, and they are meaning that they are left-handed, and this is related, as we said, to clockwise and anti-clockwise, but don't bother yourself studying this, but they are triple helices, meaning that the helices are triple, every 3 together, and this is how collagen is formed. Of course, collagen, the polypeptide chains of collagen have a specific type of amino acids, have a specific arrangement, have a specific type, arrangement, and number. But the most important characteristic is that they are triple alpha helices, triple non-alpha helices. And then we have wool, and wool consists of intertwined alpha helices intermingling together, intertwined alpha helices. Let me first erase the ink on the screen. So intertwined alpha helices like this, for example. So this is how, more or less, how it looks like. And then finally, we have silk, and silk consists of, this is the only one that consists of beta pleated sheets. But the globular proteins, that's why I was telling you that we had better postpone in the order of the slides, you'd better study fibrous and globular proteins after the tertiary and quaternary structure, because actually the primary, secondary, tertiary, and quaternary structure is present in the globular proteins. Or in other words, if the globular protein is very complicated, then it will contain polypeptide chains in various levels of complexity. So what are globular proteins? They are more or less spherical in shape, but sometimes a protein might look like a hollow cylinder, looking like the protein channels, for example, in the cell membrane. So they might be more or less spherical in shape, or they might look like hollow cylinders. So a globular protein consists of polypeptide chains, and if the globular protein is very complicated, then it will contain all levels of structure of proteins, meaning that inside the same protein, you will have a polypeptide chain that might be present in the linear form, the primary form, and that this And then this linear polypeptide chain might have been folded into a secondary structure, and then into a tertiary structure, and then came a second, meaning here you have, sorry, this is not the primary structure. So first of all, you have the primary structure, and that the primary structure has been folded into the secondary structure, and then the secondary structure has been folded more and more into the tertiary structure. And then you might also have a second type of a polypeptide chain that entered in the composition of this protein. So you might have another one. Once you have two polypeptide chains, then this is no more tertiary, but it is a quaternary globular protein. So in their highest level of complexity, the globular proteins will be in the tertiary or the quaternary structure. So this statement does not mean that inside the protein molecule you will have some primary polypeptide chains, some secondary, some polypeptide chains in the secondary level, some polypeptide chains in the tertiary level. No, but rather that it explains to you how the primary is further folded into secondary and then into tertiary. And then if the globular protein is very complex, you might have an additional different polypeptide chain that folds with the first one to form the quaternary structure. The globular proteins are essentially functional proteins. They carry out particular functions in the human body. They are usually more compact than the structural proteins, and because they are globular, then they are smaller, rather smaller or more condensed, and therefore they are more soluble. So they are functional, they are compact, they are soluble. The best group, of course, are the enzymes, and we said that they should be at least in the tertiary structure. And we also have hemoglobin. But hemoglobin is a globular protein. We have just said that it is a globular protein because it should be accommodated by the spherical red blood corpuscles. And if it loses this globular function, then it would no more be a protein. It would no more be a globular protein, and it would be it would cause sickle cell anemia. Sorry. Now we come to the tertiary structure, and the tertiary structure is a very complex 3D conformation that is superimposed on the secondary structure. And if I ever ask you, why do we say superimposed on the secondary structure? This is because it's the same molecule, the same polypeptide chain that has moved from the primary stage to the secondary level to the tertiary level. The tertiary structure, as we said before, gives the enzyme its specificity. Why does it give the enzyme its specificity? Because it allows the enzyme to be in a three-dimensional shape, and therefore you might have, as we said before, you might have depressions on the surface, and therefore you might have active sites on the surface of the enzyme, and the substrate, as you know, might come here, and or the reactant, the specific reactant that has, what is the specific reactant? The specific reactant will be that reagent or that substrate that will have a shape complementary to the shape of the active site. Now, in order to obtain the tertiary structure, we start either by an alpha helix that is further folded, or by a beta sheet that is further folded. But why are we saying this? We start because this statement is a bit ambiguous, a combination of alpha helices and beta sheets that fold as a result of non-covalent interactions. Usually it is an alpha helix. Of course, alpha helix means what? Alpha helix is a secondary structure. Beta sheet is what? Beta sheet is the other version of the secondary structure. So they want to tell you that we start by a secondary structure protein, an alpha helix, and we further fold it into a tertiary structure, or a beta pleated sheet, and we further fold it into a tertiary structure. And this is usually the case, rather than having both types, alpha and beta, together, because as we have just seen on the previous slide, for example, in alpha keratin, we have only alpha helices. In collagen, we have non-alpha helices. In wool, we have only alpha helices. So we never have them, in the famous proteins, we never had them together. We never had alpha helices and beta sheets together. So I had to explain this because the statement, the wording is a bit ambiguous. So you start by an alpha helix and you further fold it into the tertiary structure, or you start by a beta sheet and you further fold it into a tertiary structure. Of course, as we said before, in order to convert secondary into tertiary, you have to make new bonds. But what is important to know is that these new bonds are non-covalent bonds. And these non-covalent bonds will be between the R groups only. So they are between the R groups of amino acids of the same chain, or the R groups of amino acids of the same chain and the water molecule around the chain. So here we have certain important rules to reach the tertiary structure. Number one, we have to start by a secondary structure. Number two, we have to make new bonds to do further folding. So here, new bonds mean what? New bonds mean further folding. New bonds in the same chain again. New bonds in the same chain will result in what? Will result in further folding. Okay? This is the second rule. The third characteristic is that these interactions are non-covalent, and we will see how. The fourth characteristic is where the bonds are, or is related to where the bonds will be formed. They will be formed between R groups, meaning that they will not be formed between oxygen and hydrogen like in the previous examples of the secondary level, either between R groups of amino acids in the same chain or R groups of amino acids and the water molecules around the polypeptide chain. So the bonds in the tertiary structure will include hydrogen bonds. Let's see all the examples and check whether they are covalent or not. The first example, hydrogen bonding between the R groups, and we will see how. This is non-covalent, of course, or ionic bonding between R groups again. Where can we see R groups? In basic and acidic amino acids, and we will explain this. So the first group is hydrogen bonding. It's not a covalent bond. The second group is the second type is an ionic bond. Again, it's not a covalent bond. The second type of bond that might result in further folding into a tertiary structure is hydrophobic interaction. Again, it's not a covalent bond. And then the hydrophilic interaction similarly. And then the only one that might be a covalent bond is this one, the disulfide bond. And we'll be explaining all this. And then we have the van der Waals interactions. Of course, the van der Waals interactions are not covalent interactions. Let's first see how we have just proved that they are non-covalent, and we have just proved that most of these non-covalent bonds are between R groups. So what is characteristic of the bonds in the tertiary structure is that they are non-covalent bond, and that they mostly non-covalent bond with one exception, and that they occur between the R groups, and we will see how. But before seeing how, we have to discuss something very important related to amino acids. In a neutral pH, like in water, for example, if we draw the central carbon, the general formula of the amino acid, this is the central carbon atom. This is the central carbon atom. And then this is the R group. This is the H. And then this is the carboxylic group, C double bond OOH. And this is the amino group. This is the central atom because this is the carbon atom of the carboxylic group. In a neutral pH, for example, when it is put in water, what happens is that this carboxylic group will lose the hydrogen. It will lose this proton into water, and the oxygen will carry a negative charge because it lost the hydrogen. And this NH group can acquire a proton, either this proton or another proton, and we will have here this nitrogen atom carrying a positive charge because it's no more NH2, but it is NH3. So the final shape would be like this. Let's look here. So we have Cr, and then we have H, and then we have C double bond O, O negative, and then we have here NH, H, and then we have H plus. So one side of the amino acid in a neutral pH carries a negative charge, and the other side carries a positive charge. So in a neutral pH, the amino acid molecules will be polar molecules. And since they are polar molecules, they are hydrophilic molecules. They are lovers of water, and they can dissolve in water. Furthermore, this state of the amino, or the status of the amino acid where it has one negative side and one positive side is called as Zwitterion. So what is Zwitterion? It is a molecule that has one negative side and the amino acid molecule that has one negative side and one positive side. This is very important to understand how amino acids can form bonds between them if they are free in the solution. But we have to discuss something else, which is basic and acidic amino acid. Usually we have what? We have a central carbon atom, we have an R group, we have an H atom, and then we have here one carboxylic group, and we have one amino group. This is the general formula of the amino acids. And 3 items out of the 4 are known. We all know that around this central carbon atom, we always have one carboxylic group, we always have hydrogen, and we always have an amino group. But what is this R? This R might be many things. It might be CH3, for example, but sometimes it's a long chain that ends by COOH again. So in this amino acid, we will have 2 carboxylic groups. This one, number 1, and this one, number 2. Since we all know now that carboxylic groups are acidic groups or provide acidity to the amino acid, why? Because they can release a proton in the solution. So this amino acid that has in its side chain a carboxylic group, which is something exceptional and unusual, will have 2 carboxylic groups and 1 amino group. So it will have 2 acidic groups and 1 basic group. The acidic groups are the 2 carboxylic groups, and the amino group is the basic one. So it will be more acidic than basic, and this one will be considered an acidic amino acid. But sometimes the side chain does not contain COOH, but contains a second amino group. So it would be, here we have the R group, which is unknown, and then we have or variable, not unknown, COOH, and then H, and then NH2. So if this R group is a side chain that contains NH2, because every amino acid has a different R group, so we have here one acidic group that releases a proton, and this is the carboxylic group, and we have 2 basic groups. This is what we mean. Of course, this is a basic amino acid because it has 2 basic and 1 acidic. So it has more basic than acidic. Let's look at the diagram here and see the types of bonds. So, for example, here the red sphere stands for basic amino acid or basic, a basic group like the NH3 group, for example, and the off-white sphere here is acidic. So we all know that the basic is the NH3 after having acquired the proton, and that the acidic is COO after having lost the proton. So here, this is a kind of ionic bond because it is between a positively charged NH3 and a negatively charged COO. So it's like when, for example, we have a bond between sodium, a sodium ion, and a chloride ion. So this is the ionic bond. And then here we have the hydrophilic bonds. And the hydrophilic bonds are bonds that are made between polar R groups. So when we say here hydrophilic, look here, the blue ones, hydrophilic bonds between polar what? Polar R groups. So if you have an R group that does not necessarily contain COOH or NH2, but it is a polar R group, but one R group, one of these, the R group here, the variable that we don't know, has at its end a partial positive charge. And that one has this R that we don't know, and we don't want to know. It's not important to know. All what's important to know is to know that it has a partial negative charge. Then there will be this hydrophilic bond, which is essentially, it might be a hydrogen bond, but a hydrogen bond, so it is either a hydrophilic bond or a hydrogen bond between 2 polar R groups. And then we have the hydrophobic interactions when we say that the van der Waals forces result in getting the hydrophobic molecules close to each other, and they clump together away from the molecules of water. So this interaction is what we call the hydrophobic interaction. Each of these interactions will result in approximation of segments. Like here, for example, this segment of the molecule has been approximated to this segment, and therefore it results in a folding. Every time you take two different segments of the chain and you make a bond between them, then you are approximating them. The last one is the disulfide bond, and essentially it is an exception because it is a covalent bond. Sometimes there are two types of amino acids in the human body, which are cysteine and cysteine. It's difficult to discover the difference in pronunciation between the two, but actually these two amino acids contain, they are called sulfur amino acids, because they are amino acids that contain a sulfhydryl group at the end. So this is the amino acid, and it has a sulfhydryl SH group. So if this is one segment of the polypeptide chain, and this is another segment of the polypeptide chain, why am I saying this? Because in the tertiary structure, it's only one polypeptide chain, but there might be segments that come close to each other. And it happened accidentally that this sulfhydryl group and that sulfhydryl group are in front of each other. So if you remove this hydrogen and that hydrogen, the two sulfur atoms will be forced to bond with each other. So if you dehydrogenate, if you remove hydrogen, you will get this kind of bond, which is SS. And we've talked about this in human biology when we talked about curly hair and straight hair. So these are the types of bonds in the tertiary structure. I'm not going to explain the van der Waals forces again, because we did this earlier on the slide. In the quaternary structure, you have to have more than one polypeptide chain. The example of hemoglobin is a bit confusing to you at the beginning, because hemoglobin essentially consists of 4 polypeptide chains. They are 4 polypeptide chains, 2 alpha chains and 2 beta chains. And when we say here beta, we don't okay, I'm not going to go into these details, but actually the hemoglobin is made of 4 polypeptide chains, 2 alpha chains, the purple ones, and 2 beta chains, the brown ones. So it is made of 4 chains, but this is not the reason why it is called a quaternary structure. It's called a quaternary structure just simply because it is made of more than one polypeptide chain. You can see that each of these polypeptide chains is highly folded into a tertiary structure, and that at the center of each of these polypeptide chains, we have an inorganic part that is called, an inorganic iron-containing heme, meaning that heme, this is heme, this is the chemical formula of heme. Heme, of course, is organic because it contains carbon and hydrogen, but the center of it is iron, and iron is inorganic. So when we say each of the 4 polypeptide chains has an inorganic iron-containing heme group in the middle, this does not mean that the heme group is inorganic. It means that the heme group contains in its center iron, and that iron is inorganic. But heme, of course, is organic because it has the criteria of organic molecules, which is carbon, containing carbon and hydrogen. And now we move on to what we call denaturation. What is denaturation? Denaturation means that the protein changes its conformation. Let's first revise together what protein structure depends on. Of course, it depends mainly on its primary polypeptide chain, meaning the type of amino acids, their number, and their arrangement, because it is this type and arrangement of the amino acid that will determine how further folding will occur. Why? Because how further folding depends on what type of bonds will be formed, and what type of bonds depends upon what kind of amino acids are already present. So what does protein structure depend on? On the primary polypeptide chain. Why is the primary structure a major determinant in protein structure? Because it determines the number of amino acids, their arrangement, and their type, and therefore the type of bonds that will be formed between them. So folding depends on the type of bonds, and the type of bonds itself will again depend on these three criteria. But protein structure also depends on physical conditions like temperature and chemical conditions like pH and salt concentration, because every protein in the tertiary structure, especially if it is a functional protein, requires, as you know from your study of enzymes, an optimum temperature and an optimum pH to work in, and an optimum salt concentration. And if any of these is changed significantly, then this will result in a change in the bonding that occurs, and therefore a change in the folding, and therefore the protein might completely lose its function. So any unfavorable change will alter the intermolecular forces, like, for example, hydrogen bonds may be broken down, which results in a change in conformation and folding of the protein. And this is what we call denaturation. If the protein is denatured, then this results in loss of function of the protein. Sometimes this denaturation is irreversible, and sometimes it is reversible. If the change is not significant, the number of bonds that have been broken will be very small, and therefore the change in conformation will be insignificant. But if the change is significant in pH or in temperature, a greater number of bonds will be broken down, and a more significant change in shape will occur. For example, in case of boiling an egg, you all know that the white of an egg is called albumin, and that albumin, you have to know that albumin is a globular protein, and it is a soluble protein, which means that albumin may dissolve in water. But when you heat, some bonds in the globular molecule of albumin will be broken down. And when these bonds are broken down, there is a change in the structure or the conformation. So upon heating, this is the sequence that occurs. There will be bond breaking. And when there will be bond breaking, there will be conformational change in albumin. What kind of conformational change will occur, and how is it related to solidification of the boiled albumin? The conformation change is essentially an exposure of the hydrophobic amino acids, meaning that at the beginning, in the molecule of albumin, when albumin is still spherical, we only have hydrophilic amino acids at the outer surface, hydrophilic amino acids at the outer surface. And since these outer hydrophilic amino acids, or since these outer amino acids of the peptide chain of albumin are hydrophilic, then they can interact with water, because, as you know, polar likes polar. But when you heat, some bonds break, a conformational change occurs, and it results in exposure of the hydrophobic amino acids. So the hydrophobic amino acids, of course, when they are exposed, they are scared, they try to get away from the water, they try to clump together and stick to one another, and they form a network that results in this solidification pattern or status of the boiled white of the egg. Now we move on to a very important concept, which is the chaperone proteins. Usually after, we all know that, as we said before, DNA undergoes transcription into RNA, into messenger RNA, and messenger RNA will be translated into a polypeptide chain. Usually just after having been translated, these polypeptide chains will fold spontaneously into the final level of hierarchy, tertiary, quaternary, in order to be functional. And how do they fold spontaneously? Because there are certain proteins that in the cells that, and in the cells and in everywhere, when we require in the cell, in the body fluids, that result in proper folding of the protein. They help or they assist proper folding of the protein. Why are we putting all these efforts to ensure proper folding of the protein? Because if it happens that a certain protein is not properly folded or is misfolded, this can result in a serious problem of the cell and may result in, of course, why a serious problem in the cell? Because it would lose its function. This is the first reason why an unfolded or misfolded protein causes serious effects, because it would be no more functioning, and therefore the cell would lose its functions. But there is another important thing, is that these misfolded proteins might result in serious diseases like mad cow's disease. Of course, you have heard about mad cow's disease, Parkinson's disease, where we have coarse tremors of the hand, and Alzheimer's disease, of course, that results in loss of memory, and sometimes it ends by the patient ends up by having illusions and delirium, and because of degeneration and loss of function of the brain cells. These misfolded proteins are called prions. These prions or abnormal proteins are mostly dangerous in the nervous system. Why? Because cells of the nervous system, or the cell bodies of the neurons, are the site where we have the greatest number of prion proteins. So what are prion proteins? Prion proteins, if this is the cell body of the neuron, then prion proteins are normal proteins that are expressed on the cell membrane and that have very important functions in the body. But if these prion proteins, this is not polypeptide, this is prion protein, if these are misfolded, then they give rise to prions. So be very careful when you talk about prions and prion proteins, because prion proteins are normal proteins that have important functions and that are expressed on the cell membranes of most body cells, but they are mostly abundant on the cell membranes of neurons. But if they are not properly folded, they give rise to prions, okay? Prions are very dangerous. Why? First of all, because they do not carry the normal function. So just by converting prion proteins into prions, you lose the normal function of the prions. But how can we get the prions? First of all, by a mutation in the gene that encodes the protein, because prion proteins, these ones, are proteins just like any proteins in the body. and therefore they require genes or segments of DNA. So if there is a mutation in the DNA that forms the gene, then it will not be expressed into a normal prion protein, but it will be expressed into a prion. The second source is infection. But infection here is not through an infectious organism. Okay, the infection here is through the prion itself, because if you ingest, for example, the brain of an animal that is infected with mad cow's disease, therefore that contains prions, these prions are infectious, but they are infectious in the meaning that they will trigger other prion proteins that are normal to be misfolded into prions. So how are prions infectious? If they are, for example, in the brain of an animal, and some people eat brain, for example. So, or in the cell membrane in other cells, and they are ingested, they go to the blood and they are unfolded and they trigger or stimulate other normally folded prion proteins to be unfolded, making them lose their functions and result in serious diseases. But it's worth noting that if the prion itself is infectious through triggering misfolding of the prion proteins, yet the first time the prions have been formed were due to this first time was due to an original microorganism that resulted in misfolding. So here we have the PRNP, which is there are three terms to be memorized. PRNP is the gene itself that is expressed into a normal prion protein, or so we can say that the gene is expressed into, or we can say that the gene codes for. And then we have the prion, which is the infectious particle. And we said that it has been induced by an infectious organism, but the prion itself can infect other proteins by inducing them to be misfolded. What are the functions of prion proteins? First of all, they activate the immune cells, and therefore they have immune responses. They are important for communication between the cells, and we will discuss this in the second unit on the cell, because these normal prion proteins can bind with specific protein receptors on the surface of the cell and induce or stimulate an enzyme called adenylate cyclase that converts ATP into a cyclic form called cyclic AMP that is considered a secondary messenger in the cell and induces certain responses in the cell. So this is, for example, the receptor that is called a G protein coupled receptor, and we will know why it is called like this in the second unit. And the prion protein will come and bind with this receptor, and this will result in activation of a certain enzyme called adenylate cyclase, resulting in the formation of cyclic AMP. And cyclic AMP is called a secondary messenger because PRP cannot enter the cell, but it makes all the changes through cyclic AMP. So cyclic AMP is a delegate of the prion protein, and it induces a secondary response in the cell. Prion proteins are also important for the uptake of copper from the blood into the cell, and then transporting it into the cell, storing it into the cell. And copper is important because it protects against oxidation. And you know, of course, from your study of human biology, that oxidative stress is harmful to the cell. When the cell is exposed to oxidants, like, for example, ultraviolet rays, the energy of ultraviolet rays, more than its capacity to get rid of the products of oxidation. These products of oxidation will accumulate in the cell, and this would result in diminished function of the cell. They would result in diminished function of the cell. And the fourth is protection of the cell, as we've said before, from electrochemical stress. We will postpone our study of DNA until we reach the unit on DNA, but we can talk about the functional groups. We have already talked about the functional groups, and we said that they are a group of atoms that react or that give specific functions to the molecule, and they have a consistent behavior in all chemical reactions, and they are attached in organic molecules. They are attached to the carbon atom by replacing a hydrogen atom. And they're very important because they make a huge difference in the property of a chemical substance. For example, in hormones, the difference between the female hormone estradiol and the male hormone testosterone is just through the functional group. Some functional groups are polar, like the hydroxyl group. Some others are non-polar. So the hydroxyl group is polar. The methyl group, for example, the CH3, which is the methyl group, is non-polar. The carbonyl group, of course, is polar because we have a difference in electronegativity between carbon and oxygen. The carboxylic group, we said before that it might lose a proton and become negatively charged, so it is hydrophilic and charged. The amino group, we also said that the amino group can be charged and therefore make this side of the amino acid hydrophilic. The phosphate group, we said before in phospholipids that it is charged, that it releases a hydrogen ion and become negatively charged. And finally, the sulfhydryl group is polar because of the difference in electronegativity between sulfur and hydrogen. How can we visualize 3D proteins? This is not very important, but you should know X-ray crystallography, and this will be studied in full detail with the technique with the nucleic acid. Bioinformatics, of course, we can use computers and mathematical modelings in order to enter the data from analysis of the sequence of amino acids into a protein, and the computer software will tell you what is the expected tertiary structure or 3D structure of that amino acid. The last thing is the nuclear magnetic resonance, and you don't have to know the details about nuclear magnetic resonance. But as for the X-ray crystallography, it will be studied in full detail in DNA or in our study of molecular biology, and therefore I will not lengthen the video more than this. It's already very long, and by having done this, the biochemistry unit has been totally covered and in full detail. Thank you so much, and see you very soon in the second unit about the cell.